Hello everyone, my name's Iman. Welcome back to my YouTube channel. I'm so glad you're here. Today marks the start of a fully revised and updated MCAT biology series. If you followed along before, you know I'm always refining these videos to make them more structured, more clear, and of course, more comprehensive. And your feedback has played a big role in shaping these updates. Ultimately, I just want you to feel supported and confident as you study for the MCAT. Now, everything I post here is completely free because I believe that access to high quality education should never depend on your ability to pay. So in the description box below, you'll find a link to the notes as well as full transcripts of each lesson. With that, let's start with chapter 1, which is titled The Cell. In this chapter, we'll be covering the following objectives. First, we'll start with cell theory, which is a set of principles that define what a cell is and how it relates to life itself. These foundational ideas are essential to everything else we'll learn in biology. Next, we'll focus on eukaryotic cells. These are the types of cells that make up plants, animals, fungi, and protists. We're going to look at their internal structures, like their membrane-bound organelles, and we're going to discuss how the cytoskeleton helps maintain their shape and organize their contents. We're also going to talk about how eukaryotic cells come together to form tissues. After that, we'll turn our attention to prokaryotic cells. These are simpler cells like bacteria, but they're incredibly diverse and important. We'll talk about the domains they belong to, how they're classified by shape, and how they differ in their use of oxygen. We'll also go over the basic structure of a typical prokaryotic cell. From there, we're going to move into how prokaryotic cells grow and reproduce. Even though they don't divide the way our cells do, they still pass on genetic information, and they have fascinating ways of increasing their genetic diversity. We're going to talk about binary fission, genetic recombination, and the stages of population growth. Finally, we'll take a closer look at viruses and subviral particles. These aren't considered living cells, but they still play a major role in biology and in human health. We're going to examine what viruses are made of, how they replicate, and how they infect both prokaryotic and eukaryotic cells. We'll also introduce prions and viroids, which are even smaller infectious agents that disrupt biological systems in unique ways. So with that introduction in mind, let's begin by discussing the core principles of cell theory. Before the invention of the microscope, people believed that living organisms were complete and indivisible, which means that they couldn't be broken down into smaller components. But all of that changed in the 1600s when the microscope opened an entirely new world of observation. In 1665, Robert Hooke built a crude compound microscope, and he used it to examine a slice of cork. What he saw reminded him of small rooms that he called cells. Just a few years later, in 1674, Anton von Leeuwenhoek became the first person to observe a living cell under a microscope. This was a groundbreaking moment because it showed that cells were dynamic and alive. Then in 1850, Rudolf Virchow added another key insight. He demonstrated that diseased cells could arise from normal cells in healthy tissue. This suggested that cells don't just appear randomly, they come from other cells. And these discoveries led to the original version of cell theory, which includes three main tenets. First, all living things are composed of cells. Second, the cell is the basic functional unit of life. And third, all cells arise from pre-existing cells. Later, as molecular biology advanced, scientists added a fourth tenet to the theory. This final principle states that cells carry genetic information in the form of DNA. And importantly, this genetic material is passed on from parent cells to daughter cells during cell division. So together, these four principles form the foundation of our understanding of what cells are and how life operates at the microscopic level. But we'll see in our last objective that viruses challenge some of these ideas, and that's where things start to get a bit tricky, but really interesting. Now that we've established the core ideas of cell theory, we're going to begin exploring the structure and function of cells in more detail. One of the first distinctions we make among living organisms is whether they're composed of prokaryotic or eukaryotic cells. This chapter is going to cover both cell types, but we'll start with eukaryotic cells, which are the kinds found in animals, plants, fungi, and protists. Now, prokaryotic organisms, they are always single-celled. But eukaryotic organisms, they can exist as single cells or as multicellular systems with specialized tissues. And one of the key differences between them is that eukaryotic cells contain a true nucleus. This nucleus is enclosed in a membrane and it houses the cell's DNA. Prokaryotic cells do not have a nucleus. Instead, their genetic material is just found freely floating in the cytoplasm. In addition to the nucleus, eukaryotic cells contain membrane-bound organelles, which compartmentalize specific functions and allow the cell to carry out multiple processes simultaneously without interference. These organelles are suspended in the cytosol, which is a semi-fluid substance that facilitates the movement of molecules throughout the cell. The membranes that enclose these compartments, those are composed of phospholipid bilayers, which regulate what enters and exits each space. And this structural organization gives eukaryotic cells a high level of internal complexity and coordination. In this objective, again, we will focus first on eukaryotic cells. And what we're going to do is we're going to start with a broad overview of the important cellular components we need to know for the MCAT. So I'm going to introduce them in this summary table here, and then we're going to spend more time going over the nucleus, the mitochondria, the endoplasmic reticulum, and the Golgi apparatus in a lot more detail. Those are really important to master. The nucleus acts as the control center of the cell. It stores genetic information and it directs the synthesis of RNA and proteins. Mitochondria are responsible for generating energy in the form of ATP using the tricarboxylic acid cycle, also known as the Krebs cycle, and the electron transport chain. Lysosomes are organelles filled with hydrolytic enzymes that help break down waste, damaged organelles, and macromolecules. They often get this material by fusing with endosomes, which carry cargo either from outside the cell or from internal trafficking. Also beyond that, lysosomes can also trigger autolysis, where the cell intentionally releases these enzymes to break down its own components during programmed cell death, or apoptosis. Mitochondria can do this too, by the way. Then there's the endoplasmic reticulum. This exists in two forms: the rough endoplasmic reticulum, which is covered in ribosomes, and it assists in protein synthesis and processing. And the other form is the smooth endoplasmic reticulum, which is involved in lipid synthesis and detoxification. The Golgi apparatus modifies, packages, and sorts proteins and lipids for delivery to their final destinations. It also contributes to the formation of lysosomes. Lastly, peroxisomes carry out oxidative reactions, including the breakdown of fatty acids, and it detoxifies harmful byproducts. Now that we know the main components of the eukaryotic cell, let's begin discussing the structure and function of the nucleus in a lot more detail. The nucleus, again, is often described as the control center of the cell because it houses the genetic material that directs all cellular activities, from protein synthesis to cell division. The nucleus is enclosed by a double membrane structure known as the nuclear envelope, also called the nuclear membrane. This envelope separates the nuclear contents from the cytoplasm, and it helps maintain the internal environment of the nucleus. Embedded in this envelope are structures called nuclear pores. These pores are not just openings. They are highly regulated channels that allow selective two-way exchange of materials between the cytoplasm and the nucleus. So for example, messenger RNA or mRNA, it needs to exit the nucleus through these pores to reach the ribosomes, while certain proteins and enzymes have to be imported into the nucleus to support DNA replication or transcription. Now, inside the nucleus, the genetic material is organized as DNA. But this DNA is not floating around loosely. Instead, it's wrapped around proteins called histones. Together, the DNA and histone proteins form a complex called chromatin. Chromatin can be loosely packed in a form called euchromatin, which is accessible and transcriptionally active, or it can be more densely packed as heterochromatin, which is generally less active. When a cell prepares to divide, the chromatin is going to condense into visible structures called chromosomes. Each chromosome is made of a single continuous molecule of DNA, tightly wound and organized for efficient separation during cell division. And we're going to cover that in a lot more detail in chapter 2. Within the nucleus, there is also a dense membraneless region called the nucleolus. This is not involved in storing DNA, but rather in producing ribosomal RNA or rRNA. This type of RNA is a key structural and functional component of the ribosome. And the nucleolus assembles rRNA with ribosomal proteins to form the subunits of ribosomes, which are then exported to the cytoplasm where they become active in protein synthesis. Finally, surrounding the DNA and the nucleolus is the nucleoplasm. This is a viscous, gel-like substance that supports nuclear structures, and it allows molecules like nucleotides and enzymes to diffuse throughout the nucleus. So altogether, the nucleus integrates genetic storage, regulation, and ribosome production into one well-organized structure that makes it really essential for the proper functioning and survival of eukaryotic cells. Next, we want to cover mitochondria. These organelles are best known as the powerhouses of the cell, and that's because they're the primary site of ATP production. But beyond energy generation, mitochondria also have several unique structural and genetic features that make them especially important to understand. Mitochondria are enclosed by two membranes. You have the outer membrane, which separates the mitochondrion from the cytosol, and it acts as a boundary between the cell and the internal mitochondrial environment. It's relatively porous, and it allows small molecules and ions to pass freely into the space between the membranes. This region is called the intermembrane space. The inner membrane, in contrast, this is highly selective and much more specialized. It is folded into numerous infoldings called cristae. These serve to increase the surface area available for energy-producing reactions. Embedded in this inner membrane are the components of the electron transport chain. This is the final step of aerobic respiration, where ATP is synthesized using the energy carried by electrons. The folding of the cristae provides more space for these reactions to occur efficiently. Enclosed by that inner membrane is the mitochondrial matrix. The matrix contains enzymes of the citric acid cycle or Krebs cycle, as well as mitochondrial DNA and ribosomes. This is where several metabolic reactions take place, and it's also where mitochondrial replication and gene expression are coordinated. What makes mitochondria especially interesting is that they are semi-autonomous. Unlike most other organelles, mitochondria contain their own circular DNA and ribosomes, which allows them to transcribe and translate some of their own proteins. They are able to replicate independently of the nucleus through a process called binary fission. We're going to cover that in a later objective in this chapter. Because mitochondrial DNA is separate from nuclear DNA, it is inherited through a process known as cytoplasmic or extranuclear inheritance. In most organisms, mitochondrial DNA is passed down exclusively through the maternal line. And that's because during fertilization, the mitochondria from the sperm are typically not retained in the embryo. Now, this unique feature of mitochondrial genetics is part of the evidence supporting the endosymbiotic theory. And according to this theory, mitochondria are thought to have originated when ancestral eukaryotic cells engulfed an aerobic prokaryote. And rather than being digested, the two cells formed a mutually beneficial relationship. They were friends with benefits. And then over time, the engulfed prokaryote evolved into the mitochondria we see in modern cells today. So those friends with benefits finally got married. So in addition to being the center for energy metabolism, mitochondria also offers some really important insight into the evolutionary history of eukaryotic life. Now, let's move on to the endoplasmic reticulum, or ER. The ER is a continuous membrane system that plays a central role in the synthesis, folding, modification, and transport of cellular materials. Now, structurally, it consists of a network of interconnected membrane-bound sacs and tubules, and it's actually physically connected to the outer membrane of the nuclear envelope. Now, this continuity allows for direct movement of materials between the nucleus and the ER. There are two types of endoplasmic reticulum, and each one has a distinct structure and function. We have the rough ER and we have the smooth ER. The rough ER, it gets its name from the ribosomes that are attached to the surface. These ribosomes, they're not permanent fixtures. They bind when the cell is actively translating proteins that are meant to be secreted, inserted into the membrane, or delivered to organelles. And as proteins are synthesized on these ribosomes, they're threaded directly into the lumen of the rough ER, where they undergo folding and some post-translational modifications. In this way, the rough ER serves as a major hub or site for protein synthesis, especially for proteins that are destined to leave the cell or be embedded in membranes. Now, in contrast to that, the smooth endoplasmic reticulum, it lacks ribosomes, as you can tell. And there's no ribosomes on the surface. It's therefore not involved in protein synthesis. Instead, it serves other functions. It's involved in lipid synthesis, including the production of things like phospholipids and steroids. It also plays a role in detoxifying drugs and poisons, especially in liver cells. And in certain cells, it can help regulate calcium ion storage and release. Now, while the smooth ER does not synthesize proteins itself, it does play a key role in transporting proteins that were synthesized by the rough ER. And these proteins are packaged and directed towards their next destination, which, you guessed it, is the Golgi apparatus. The Golgi apparatus is a critical component of the cell's transport and modification system. Structurally, the Golgi apparatus consists of a series of flattened, membrane-bound sacs called cisternae. These sacs are stacked together, and each one has a distinct role in processing cellular products. The Golgi also has a defined polarity. The cis face is the one that receives incoming vesicles, and the trans face is where modified products are sent out. Vesicles from the smooth endoplasmic reticulum, they fuse with the cis face, and they release their contents into the lumen of the Golgi apparatus for modification. Inside the Golgi, proteins and lipids can undergo various modifications. This includes glycosylation, where carbohydrate groups are added to proteins, as well as things like phosphorylation and sulfation, which can affect how the molecules function and where they're targeted. Now, once they're modified, the products are repackaged into new vesicles that bud off from that trans face of the Golgi. These vesicles are going to be directed to specific destinations within the cell. So some may be sent into the lysosomes or to the plasma membrane, while others are just going to be transported out of the cell entirely. If a protein is destined for secretion, the vesicle carrying it is going to merge with the plasma membrane in a process known as exocytosis that releases those contents into the extracellular space. With the ER and the Golgi working together, the cell is really able to efficiently synthesize, modify, sort, and deliver a wide range of biomolecules. Now that we've covered how molecules are transported and processed within and inside the cell, let's shift our focus to another key system that supports cellular structure and movement, and that's going to be the cytoskeleton. The cytoskeleton gives the cell its shape and its structural support. But it doesn't just hold the cell together. It also plays a very important role in things like transport and in movement and even cell division. The cytoskeleton, it's made up of three main types of protein-based filaments. We have microfilaments, microtubules, and intermediate filaments. Each type has its own structure and function. and we're going to go over each in detail. We'll start with microfilaments. These are the thinnest filaments in the cytoskeleton, and they're made of a protein called actin. The actin molecules are polymerized into solid rods, which are usually arranged in tight bundles just beneath the cell membrane. These filaments help the cell keep its shape by resisting external pressure or compression. Now, microfilaments aren't just static. They're dynamic and they're involved in movement. When they work together with a motor protein called myosin, they can use ATP to generate force for movement. And this actin-myosin interaction is how muscle cells contract. But it also plays a role in things like cell crawling or changing shape. Another important function of microfilaments is during cell division. In the final step, called cytokinesis, actin filaments form a ring around the middle of the dividing cell, and this ring tightens and eventually pinches the cell into two new daughter cells. Next up is microtubules. These are much larger than microfilaments, and they're shaped like hollow tubes. They're made from polymerized subunits of a protein called tubulin. In microtubules, they stretch throughout the cell, and they kind of act like internal highways for transporting materials. Specialized motor proteins like kinesin and dynein, they walk along these microtubule tracks, and they carry vesicles and other cargo to where they're needed. Microtubules are also responsible for forming motile structures like cilia and flagella. These structures, they both use the same internal framework, a 9+2 arrangement of microtubules. So that means there are 9 pairs of microtubules arranged in a ring with 2 additional microtubules in the center. Now, cilia, these are short projections that move fluids or particles along the surface of the cell. So for example, the respiratory tract, it's lined with cilia to move mucus upward. Flagella, in contrast, they're longer and they're used to move the cell itself. So sperm cells are a good example of that. During cell division, microtubules can also organize into the mitotic spindle, which is what separates the chromosomes. So this organization starts in a region called the centrosome, which contains a pair of structures called centrioles. And what happens here is the following. Each centriole, it's made of 9 triplets of microtubules arranged in a ring. The centrioles help organize the spindle fibers, which then attach to chromosomes at specific sites called kinetochores. And this connection is essential for making sure each daughter cell gets the right number of chromosomes. Now, this is a very quick explanation. We're gonna go into details about this in chapter 2. But keep in mind that microtubules are important for this process. They're important for cell division. Finally, we have intermediate filaments. These filaments are a bit more stable and they're less dynamic than the other 2 types. Their main job is really just to provide mechanical strength. They help the cell hold its shape. They help the cell resist stress and keep internal structures in place. They're also really important for anchoring organelles and maintaining connections between neighboring cells, especially in tissues that experience mechanical strain. What's really interesting about intermediate filaments is that their composition varies depending on the cell type. And because of this variability, intermediate filaments are often used as molecular markers to identify different tissue types. And that brings us to the final topic for Objective 2, which is tissue formation. One of the defining features of eukaryotic cells is their ability to organize into complex tissues. This allows for division of labor, where different cells within a tissue take on specialized roles. So instead of every cell doing everything, tasks are distributed, and that makes multicellular organisms more efficient and more adaptable. Now, there are four major types of tissue in the human body: epithelial tissue, connective tissue, muscle tissue, and nervous tissue, and each plays a distinct role. Muscle and nervous tissues are going to be covered in later chapters, so today we're just going to focus on epithelial and connective. Epithelial tissues cover the body and they line internal cavities. The epithelial cells are tightly packed together and they rest on a thin supporting layer called the basement membrane. This basement membrane provides structural support and it acts as a barrier between the epithelium and underlying tissues. In many organs, epithelial cells form the parenchyma. This is the functional part of the organ, the part that actually carries out its main physiological role. So for example, in the kidney, the epithelial cells of the nephron are responsible for filtering blood and forming urine. Epithelia can also be classified based on both the number of layers and the shape of the cells. If the tissue has only one layer of cells, it's referred to as simple epithelium. If it has multiple layers, it's referred to as stratified epithelium. There's also a special case called pseudostratified. This type of epithelium appears to be layered when viewed under the microscope, but actually all of the cells are in contact with the basement membrane, so it's technically only one layer. Then, in terms of shape, epithelial cells can be cuboidal, which is cube-shaped. They can be columnar, which are tall and more rectangular, or they can be squamous, which is flat and scale-like. These structural differences influence how the epithelium functions. So for example, squamous epithelium is ideal for diffusion, while columnar epithelium is more specialized for absorption and secretion. Now let's turn to connective tissue. Unlike epithelial tissue, which forms coverings and linings, connective tissue provides support and structure. Its primary role is to form the stroma, which is the supportive framework that holds organs together and provides a place for other tissue types to anchor. Most connective tissue cells produce and secrete proteins like collagen and elastin, and these proteins make up the extracellular matrix. This is a network of fibers and ground substance that gives connective tissue its strength and its elasticity. The extracellular matrix is what allows tendons to resist tension. It's what allows skin to remain flexible, and it's what allows cartilage to cushion joints. So together, epithelial and connective tissues work in tandem. Epithelial cells carry out essential functions, while connective tissues provide the support and the infrastructure needed to keep those functions stable. And understanding how these tissues are built and how they interact lays the groundwork for studying organ systems and diseases later in this playlist. For now, we're going to go ahead and wrap up the first part of this chapter. In the next video, we'll continue and finish chapter 1. Please let me know if you have any questions, comments, concerns down below. Other than that, good luck, happy studying, and have a beautiful, beautiful day, future doctors.